Okay, so it is my pleasure to introduce this year's winner of the Elizabeth D. Hay Award. Since 2017, the SDB has made this award to an investigator who's early in their independent career, performing original, significant research with impact on the broader field of developmental biology. This is actually my favorite award that we give because it's forward-looking and looking to what the future of the field is. And given the amazing nominees that we had this year, the future of developmental biology is not only strong, it's incredibly exciting, and it's going to be great to watch it unfold in the years ahead. There were numerous deserving nominees this year, but I'm very pleased to introduce this year's winner, Dan Dickinson, who is an assistant professor at the University of Texas at Austin. So Betty Hay, who this award was named for, was a woman of many firsts. In 1973, she was the first woman to be elected president of SDB. She was also the first woman to be elected president of the American Society for Cell Biology. She was the first woman to win our Conklin Medal. Scientifically, she was the first person to show that the extracellular matrix plays a vital role in determining cell behaviors. And she's probably best known for her research in limb regeneration, the role of the ECM in cell differentiation, and of course, epithelial to mesenchymal transitions. And I have to say that I was fortunate enough to know Betty and be mentored by her a bit when I was a graduate student. And she was just a truly inspiring person and a real pioneer, and really one of the only a handful of women faculty in the sciences at Harvard when I was there as a graduate student. This award is given in her honor, and it's well-deserved. Okay, so Dan. Dan did his PhD working at Stanford with James Nelson and Bill Weiss. His beautiful thesis work was on alpha-catenin and epithelial cell polarity and dictyostelium. He was unbelievably productive and published 5 papers, including a first author science paper and 2 first author Dev Cell papers. So then for his postdoctoral work, he moved to UNC Chapel Hill to work with Bob Goldstein, and obviously switched to working on C. elegans, which he's been working on ever since. He was even more productive as a postdoc, authoring 13 papers. But perhaps more importantly, this is where he started on the path of really developing transformative technologies at the intersection of cell biology, developmental biology, and biochemistry. He was actually at the forefront of adapting CRISPR technologies to work in C. elegans. In just 1 year after Cas9 was demonstrated to function as an RNA-guided exonuclease in the test tube, Dan managed to use it to engineer the genome of C. elegans. He also showed that it could be used not only to knock out genes, but to knock in precise modifications into the worm genome. As an independent investigator, Dan has published 14 research articles, on 10 of which he's corresponding or co-corresponding for author. Since starting his lab at UT Austin works on mechanisms that polarize the first cell in the embryo, and the central question is, how do the molecules that function at the nanoscale create patterns at the micron scale? And traditionally, molecules like this have been studied using biochemical approaches in vitro, but a huge limitation of that is that the molecules are being examined outside their natural environment in buffers that only partially approximate how things are in vivo. And so Dan devised new methodologies to address this challenge head-on. He combined established single-cell microscopy techniques with ingenious new methods to label and observe molecules in native cytoplasm. And as you can see here, he really brings a sense of wonder, not just to his science, but really to everything he does, even his dinner. So, please, Dan is an active participant in our developmental biology community. He's taught in the embryology course at Woods Hole and has also served as a TA in the physiology course. He's the recipient of an NSF Career Award and has also won teaching awards and best paper awards, and is a talented and dedicated mentor. Please join me in congratulating Dan on this well-deserved award and welcome him to give his talk.
All right. Thank you, Carole, for that really nice introduction, and thank you to the Society and all of you. When I was first nominated for this award, I spent some time looking over the people who have gotten it in the past, and it's a tremendous group of people who I really admire. And so to be part of that group really just leaves me speechless. And so I'm so grateful to the selection committee and the Society and all the people that nominated me, and very excited to tell you a little bit about our work today. Before I dive into the science, I want to just take a moment to celebrate my lab as it's grown over the years. This really is just a phenomenal group of people, graduate students, techs, postbacks, undergrads, and my kids are in this picture too. You know, this award is a celebration of their work as much as it is mine. I'm just so grateful to them, not only for their hard work, but also for their creativity and ingenuity, and also just for being delightful people who make me excited to come to work every day. Just a huge thank you to them for everything that they've done. I will show you work today from Nadia Manzi and Naomi Stolpner and Yash Shukla and Shangping Su, 4 grad students in the lab, along with Lars Deutz and Sena Sarakaya, who are undergrads and postbacks in the lab. So our work has focused on cell polarity, which is one of the fundamental eukaryotic cell behaviors that enables development to occur. Developing cells or cells in a developing embryo need polarity in order to assemble functional tissues, to carry out tissue shape changes, to migrate and find where they're going, to divide asymmetrically. This, by the way, is the second time you've seen this picture today. It was in Chris's talk as well, one of my favorite pictures of neuroblasts on the far right there. And then finally, they need polarity to actually form cells that can carry out functions in vivo. And one of the remarkable things about animal cell polarity is that there's a single group of proteins called the partitioning proteins, or PAR proteins for short, that play a role in polarizing many distinct types of cells. So they're involved in apical basal polarity in epithelia, in axon specification in neurons, in asymmetric cell division, both in the C. elegans zygote, which is the cell that you see here, as well as in many other asymmetrically dividing stem cell types, and in migrating cells, both single and collective migration. The core of this protein system is a pair of protein kinases called APKC and PAR1 that are found on opposite sides of a polarized cell. And once they get there, they can phosphorylate downstream components that actually endow cells with polarized cell behavior. So these are things like microtubule motors, cell fate determinants, components of the actomyosin machinery. Now, these kinases don't get where they go on their own. They're recruited by a whole alphabet soup of other partner proteins, which I won't ask you to memorize on the spot. But this really raises the question of what are the signaling complexes that are assembled that generate asymmetry? So how do you actually take this constellation of factors and put together molecular machines that can break symmetry and polarize a cell? The second thing that really fascinates me about this system is that it can function to polarize cell types that superficially look very little alike. So you would never look at a neuron and a C. elegans zygote and go, oh, I bet those cells are using the same machinery to polarize. But in fact, they are. And so that suggests that somehow developmental signals impinge upon this machinery to remodel it or reprogram it in a way that allows it to function in different contexts. And so we'd like to understand not only how are signaling complexes made and used by cells to break symmetry, but how are those cells responsive to developmental signals to allow them to function in an appropriate context-specific way. Now, if you think about it for a moment, there's a really deep tension in these two questions that I've posed, because thinking about how proteins interact and form signaling complexes is a fundamentally biochemical question. And yet, I'm asking questions about development, which fundamentally cannot be addressed in vitro in a test tube. And so this tension really bothered me throughout my graduate training, and it's motivated me to try to develop new tools that will allow us to ask biochemical questions in a more in vivo context in a developing embryo. So here's what we've done. So we start by labeling the proteins that we want to study using CRISPR. Carole mentioned I put a lot of work into getting this to be really efficient in C. elegans and more recently in embryonic stem cells. Then we take a cell, in this case a C. elegans zygote, we put it in a microfluidic channel and we explode it using a shot from a pulsed infrared laser. So this generates a cell lysate, or an embryo lysate, in a volume of just a couple nanoliters that's encased in this microfluidic device. Now, we can't suck this lysate out and run a gel on it. There's not enough. And so instead, we build these devices on top of glass cover slips that are derivatized by antibodies. So we can immunoprecipitate tagged proteins onto the surface of the cover slip. And we do this whole experiment on top of a turf microscope so that we can watch that immunoprecipitation happen in real time and detect and count the protein complexes that come down. So this is what the data actually look like. Here we've exploded a cell carrying some labeled protein complexes just off screen, and we see this beautiful shower of single fluorescent molecules landing. Now, each of these dots is a single protein complex, and if you look carefully, you'll notice that most of them are white when they first appear. That's because green and magenta are appearing together. Those two proteins are interacting. After that, some of the labels disappear due to photo-bleaching, and so that's why you see some green and magenta flickering as well. So we've written software to detect and count many thousands of complexes per cell, per experiment. The whole experiment takes about 10 minutes from start to finish. So it's a very rapid and high throughput way of asking questions about protein interactions in vivo. So this allows us to study the composition of protein assemblies and how these change between cells and over time, because we start with single cells that we can precisely stage. It allows us to measure interactions between any pair of proteins we can tag, because we use the same 3 antibodies for every experiment, which recognize the fluorescent tags that we knock into the genome. We preserve native expression levels by labeling with CRISPR, so we don't have to worry about overexpression. Since we're counting single complexes, we can think about heterogeneity between complexes involving the same protein in the same cell. And as a side benefit, this allows us to work with proteins that are difficult or impossible to purify, which happens to include almost all the ones we're interested in. I'm going to tell you a story today that I think illustrates the power of this approach and how we've used it to dissect some polarity machinery in the zygote. But I really want to emphasize the generality of this technique. We happen to be working on PAR proteins, but we could be working on anything that we can label. Okay, so we're going to focus today on the development of the C. elegans zygote. The anterior-posterior axis in this animal is established at the 1-cell stage when the zygote divides. So every single cell goes through a polarity establishment phase in which this group of 3 proteins, including our favorite kinase APKC, gets segregated to the anterior. And then polarity is maintained for a few minutes before the cell divides. If we look at PAR3, which is a scaffold protein that recruits our kinase APKC to the membrane, it forms these clusters on the cell surface. These are actually oligomers that are segregated towards the anterior by actin-myosin flow as the cell polarizes. We know that oligomerization is important for this segregation because if we make a monomeric mutant that can't oligomerize, the cells can no longer polarize. And so the model when we started, which came from some of my work, but much more work from Ed Monroe and Nate Gehring's lab and the Rodriguez lab and others, was that PAR3 recruits APKC into clusters and carries it to the anterior side of the cell. So what we wanted to know is, how do these clusters actually assemble? How do they recruit APKC? So we started by just breaking open C. elegans zygotes at different stages. And you can see right away that we can capture these really large clusters of PAR3 from establishment phase zygotes, and only from establishment phase zygotes. So this is a period of time about 7 minutes long during which oligomerization occurs, and we can see that it's developmentally regulated. Now, we can quantify this by using single molecule photo-bleaching step counting to actually count the number of molecules in each of those dots. And since PAR3 forms linear oligomers, as you can see up there on the top left, or top right for you, I guess, the oligomerization is linear, and the distribution of sizes that we expect for a situation like that is an exponential distribution. That's because every oligomer, regardless of its size, has the same probability of adding another monomer. And so that gives rise to an exponential curve. However, when we actually measure the distribution of PAR3 oligomers in zygotes, we find a distribution that's skewed from an exponential. And in particular, what I want to draw your attention to is that the largest oligomers, those with more than about 5 subunits, are overrepresented by many orders of magnitude compared to a simple exponential model. Now, I'm going to show you a lot of graphs like this. On the x-axis, you're going to see oligomer size, the number of molecules per complex, and on the y-axis, the abundance on a logarithmic scale. And pay attention to this sort of biphasic behavior. That's what we're going to pay a lot of attention to through the rest of the talk. That is indicating the overrepresentation of these larger clusters. Okay, so large oligomers are much more abundant than expected. Why? Well, we found out when we labeled PAR6 and APKC. And what I hope you can see from this image is that when we co-label these two proteins, only the larger clusters contain PAR6. Again, we can quantify this by counting the number of molecules per complex and then just asking whether those complexes contain the kinase or not. And regardless of whether we label APKC or PAR6, which is its cofactor, we get the same result, which is that there's a sharp increase in the abundance of interaction with PAR6 and APKC at a threshold size of about 3 monomers. Now, we can do this analysis in a slightly different way. If I bring back that size distribution plot I showed you a moment ago, what I'm going to do now is separate the population of PAR3 oligomers into complexes that contain APKC and those that don't. And what you can see is that the complexes that contain APKC, in green here, fall along an exponential distribution with a shallower slope than those that don't contain APKC. In other words, those that contain APKC oligomerize more strongly than those that don't. So, oligomerization of PAR3 promotes binding to APKC. That's the graph on the left. The presence of APKC shifts the distribution of oligomer sizes larger. That's on the right. These two factors reinforce one another. That's another way of saying that they're biochemically cooperative. So PAR3 oligomers cooperatively recruit PAR6 and APKC to assemble these large clusters that move towards one side of the cell. I won't show you the data in the interest of time, but these complexes are really stable, and this size threshold corresponds to what we know to be the minimum size that allows segregation by cortical flow. So how does this happen? Our working model is that PAR3 and APKC form multivalent complexes, that each APKC monomer touches directly or indirectly more than one PAR3 molecule within a cluster, thereby cross-linking and stabilizing the assembly. And we've tested this by using CRISPR to engineer mutations into the endogenous proteins in C. elegans. So we took advantage of some really nice in vitro work from Ken Prahoda's lab. They identified a PDZ ligand at the C terminus of APKC that binds to a PDZ domain on PAR3. And when we mutated that, and then looked at the distribution of PAR3 oligomer sizes, we find that overrepresentation of large oligomers is gone. The distribution is now shifted much more closely towards the exponential that we expect for simple oligomerization. So we've broken the cooperativity. However, when we look at the abundance of PAR3 APKC complexes, we find that it's reduced much less. And in particular, if you focus on the monomer and dimer, the first two data points in this graph, you'll see that they're almost unchanged in abundance compared to the wild type. So even though we've broken the cooperativity with this mutant, we haven't broken the interaction. That implies there must be another binding site. And what is the other binding site? Well, it's probably PAR6, which is also reported to have interactions with PAR3. And sure enough, when we deplete PAR6 with RNAi, we see exactly the same thing. We reduce the cooperativity, but we don't completely break the interaction. So this demonstrates that there are multiple binding sites between PAR3, PAR6, and APKC in vivo, and those multiple interactions create cross-linked assemblies that form oligomers that can segregate. Now, even though any single change doesn't strongly disrupt the association between PAR3 and APKC, it does profoundly disrupt cell polarity. So this is back to the PDZ ligand mutant. In this mutant, PAR3 can't get to the membrane, it can't recruit PAR6, the cells can't polarize, and they divide symmetrically and die. This embryo on the bottom left there, or sorry, bottom right, I keep forgetting that you're backwards. This embryo on the bottom right does not go on to make a worm, it goes on to make a pile of the wrong kind of cells. And so this cooperative assembly of PAR3 complexes is critical for partitioning and embryonic development. Okay, so what happens when polarity establishment is over, right? I mentioned that we only saw those big complexes when we broke open establishment phase embryos. And in fact, at the end of polarity establishment, when polarity is stably maintained, PAR3 actually declusters and leaves the membrane. And when we do single molecule pull down experiments at that stage, we find that the cooperativity is largely gone. So wild type PAR3 during maintenance looks a lot like our mutants during polarity establishment. Despite this, APKC remains at the plasma membrane. So that implies that something else must be recruiting it there. And the something else is almost certainly CDC42, which is right here in this cartoon, right? I don't think I can point from across the room, but over here. Not too bad. So CDC42 is a small GTPase. It's a member of the Rho family, and it has known interactions with PAR6, and it's known to be genetically essential for polarity maintenance. So we wanted to test this interaction biochemically. The challenge is CDC42 is a lipid-anchored membrane protein. We never saw it binding to PAR6 or APKC, or for that matter, anything else when we broke open cells in detergent. So the solution to that was to adapt what are called lipid nanodisc forming polymers. So for those of you who haven't seen these before, they were developed originally for doing cryo-EM on membrane proteins. And what they are is amphipathic polymers that intercalate within the membrane, and they punch out these beautiful little Oreo cookies of membrane, native cellular membrane with proteins intact inside them. And so we blew open some cells in the presence of these polymers. We found that we could solubilize CDC42 from the membrane within just a few seconds. And we know we're forming lipid nanodiscs because we can suck our lysate out of the device and look at it on negative stain EM. And there they are, little beautiful membrane islands containing CDC42. So most excitingly, that allowed us now for the first time to actually detect native CDC42 PAR6 interactions. And for this experiment, we measured the stability of those complexes by looking at how long PAR6 molecules stay bound to CDC42 after capture. So by measuring a distribution of dwell times across hundreds of molecules in dozens of single-cell experiments, we can calculate a K-off of the interaction. So this actually gives direct thermodynamic information about the stability of that complex in vivo. And when we measured that for establishment versus maintenance phase embryos, we saw something really striking, which is that CDC42 and PAR6 bind more strongly during maintenance phase than during establishment. The off rate of the interaction is slower. That's really striking for two reasons. First of all, we know that CDC42 is genetically dispensable during establishment when it binds more weakly to PAR6, and we know that it's genetically required during maintenance when it binds more strongly. So that makes a lot of sense. The other reason this is striking is because there's, again, only about 5 minutes between these two stages of development. So the cell is really rapidly remodeling the binding affinity between these two proteins. That almost certainly required some kind of post-translational modification. So we collaborated with Josana Rodriguez's group at the University of Newcastle. They identified a phosphorylation site on CDC42, which is actually a target of APKC itself. And so we made phosphomimetic mutations at that site. And strikingly, we found that a phosphomimetic mutation at that serine 71 site increased the off rate. It destabilized the interaction, suggesting that phosphorylation on that residue actually weakens the interaction between CDC42 and PAR6. And this is just a correlation, but the off rates we observe are very similar to the developmental differences that we see between polarity establishment and maintenance, suggesting this might explain how that developmental regulation is occurring. Okay, so I'm going to stop with the biochemistry now, but I want to just take a moment to pause and point out the fact that we can now use mutagenesis in living embryos to pick apart protein complexes and how they're remodeled during development, which I think is really exciting. Okay, so I want to turn now to the second question that I posed, right? How are complexes remodeled to achieve different outcomes in development? And so we've begun to approach that question by thinking about the subsequent development of the C. elegans embryo. In the zygote, as I've already shown you, APKC and its friends are localized towards the left side of the embryo. I got the direction right this time. The future anterior side of the embryo. But just about 30 to 45 minutes later at the 8-cell stage, they're localized to the outside contact-free surface of the embryo. So they're switching from an anterior-posterior to an inside-outside polarity. And they actually form these really nice little apical caps on the somatic blastomeres. This was first shown by Jeremy Nance in the early 2000s, and we published a paper on this last year in Current Biology that you can go and look at if you're curious. So I'm just going to summarize the results of that paper, and then I want to get to some unpublished findings. So first of all, what we did was to just compare how does polarity at the 8-cell stage compare to what we know about polarity in the zygote? So first of all, we found that at both stages, at the 8-cell stage as well as in the zygote, PAR3 recruits PAR6 and APKC into clusters. And you can see them at the apical surface in these images. However, that's where the commonalities ended. So in the zygote, I already told you, actomyosin flows are required to polarize PAR3 and PAR6 and APKC. That's that dramatic movie where they carry the clusters towards one side of the cell. We found that although flows happen at the 8-cell stage, they're not required for polarity. We could block them completely with a myosin TS mutant, and we still got totally normal-looking apical caps. Unlike in the zygote, we found no evidence for a role of CDC42 at this stage. It seems to be dispensable for polarity. We also found that PAR1 does not seem to be required to polarize APKC. So this idea of mutual antagonism that's become the dogma in the field based on genetics in the zygote seems not to hold at the 8-cell stage. So what are the differences between these stages? Well, one obvious one is cell-cell contact. So we thought, perhaps cell-cell contact is the key to understanding the difference. And indeed, that's consistent with Jeremy's work. They'd found some contact-localized proteins that play roles in polarity. So we started by looking at intact embryos, and then following a cue from my postdoc advisor, Bob Goldstein, we pulled the cells apart. So here in the left, we've got 4 out of the 8 cells in the embryo. And you can see they all form apical caps on the outside, away from the center. In the middle there is a pair of cells, 2 out of the 8 cells, and you can see they each form polarized domains on opposite sides from the cell-cell contact. But what really shocked us was when we got just 1 cell, it was still able to polarize. So even though cell-cell contacts play a role in orienting polarity, they're not required for the establishment of polarity. There's some cell-intrinsic mechanism that's breaking symmetry in these cells that's responsive to cell-cell contact. So this leaves us with a conundrum, right? Apical basal polarity looks really different from zygote polarity, despite only 30 minutes between these stages. But we're now left confused about how PAR3 might sense cell-cell contacts and how this different mechanism of polarization might be established at the 8-cell stage. So we've started to think about how this remodeling might occur. We know from some of Jeremy's work in the early 2000s that this does not require zygotic transcription. You can block it with transcription inhibitors, and the cells still establish apical-basal polarity just fine. So we started thinking about new protein translation. C. elegans, just like most other embryos, is chock full of maternal mRNAs. And those maternal mRNAs are regulated by mRNA binding proteins that interact with 3' UTRs and shut down translation. One of those proteins is called OMA1. It's an RNA binding protein that blocks translation of maternal RNAs. OMA1 is proteasomally degraded, starting at the 2-cell stage and complete by the 4-cell stage, as you can see in these images. And that frees its targets to be translated by the ribosome and produce new proteins. So we got our hands on an allele of OMA1 that cannot be degraded. We call it persistent OMA1. And in those embryos, the cells at the 8-cell stage are completely unable to polarize and form apical caps, even though zygote polarity is just fine. So this leads to a model whereby OMA1 degradation licenses translation of new polarity components that reprogram the machinery and allow it to adopt an apical-basal orientation. So to identify those proteins, we've turned to ribosome footprinting. So our colleague at UT Austin, John Jenick, has come up with ways of doing ribosome footprinting on low numbers of cells. And so what we've done is to isolate embryos of different stages, perform RNA-seq, and then perform ribosome footprinting. For those who aren't familiar with this technique, basically what you do is pause all the ribosomes with puromycin, digest all the mRNA with RNases, and then you sequence the little 30 nucleotide fragments that are left behind because the ribosome is stuck to them and protecting them from the RNAs. And from those data, you can work out how many ribosomes are on each transcript, which is called translational efficiency. So we've done this now for early C. elegans embryos. And the first thing that we found, which is really striking, if you focus on the top row of graphs here, is that the correlation between RNA abundance and RNA translation in these embryos is really poor. And this is not an artifact of our data. We've done all the quality control. The data are good. This is just the truth for these early embryos. Unlike in most cell types, there are many RNAs that are around but are not being translated. Now, in the bottom half of the slide, when we compare 1-cell and 4-cell embryos, we find very few changes in mRNA abundance, but many changes in ribosome occupancy and many changes in translational efficiency. And so if we now just focus on translational efficiency and we focus on the upregulated genes, we find that comparing wild type to OMA1 mutants, most of the genes that are upregulated in wild type are upregulated less in an OMA1 mutant. So that suggests that OMA1 degradation is licensing the translation of new components. And we're now very excited to know what those components are. This is ongoing work. So the takeaways from this are that embryos can rewire developmental signaling pathways, including polarity, by adding new components. We're in a good position now to figure out which components are the key ones, and then, of course, beyond that, how it works mechanistically. So I will leave you with that thought, and I want to once again thank the group. I want to also thank our collaborators, my mentors throughout the years, especially my graduate advisors, Bill Weiss and James Nelson, my postdoc advisor, Bob Goldstein, and John Wallingford and Steve Vokes, and the other folks at UT Austin who have really helped me grow into this role. And finally, of course, our funding sources. Everybody always thanks their funding sources. I want to be clear that I really mean it. NIGMS has been incredibly generous to me over my career and helping me get started. We really sometimes lose sight of the fact that it really is a privilege to be able to do this work and to have the support of these funding agencies to do what we do. Okay, so I will stop there. I guess I don't have time to take questions, but in lieu of questions, I'll just leave you this. If you want to blow up cells, it's really not that hard. Click on the QR code or come and talk to me. Thank you.